Ever wondered how the miracle of life unfolds? How DNA replication begins? Each moment, this complex process is happening within the cells of all living organisms. It's a marvel of nature's engineering, a dance of molecules that ensures life continues from one generation to the next. At the heart of this process, we find DNA replication, an intricate mechanism that duplicates the genetic material, ensuring that each new cell carries the same genetic information as its parent. But like any well-oiled machine, DNA replication involves a host of specialized components, each with a unique and vital role. One such key player is an enzyme known as DNA primase. This tiny molecular machine is responsible for synthesizing short stretches of RNA sequences, called primers. These primers play a crucial role in initiating the addition of nucleotides, the building blocks of DNA, setting the stage for the replication process. Intriguing, isn't it? But how does this DNA primase fit into the picture? Let's dive in. DNA primase, an enzyme, is the unsung hero in the DNA replication process. Let's delve into its role and see how it sets the stage for life's most intricate dance, DNA replication. DNA primase is like a master composer, uniquely capable of synthesizing short stretches of RNA sequences. But these aren't just any sequences. These are primers, the vital starting points for the addition of nucleotides, the building blocks of DNA. Now, picture DNA polymerase, the diligent builder in the replication process. It's ready to add nucleotides, but there's a catch. DNA polymerase can only add nucleotides at a specific spot, a pre-existing 3-hydroxyl group. This is where the role of DNA primase becomes crucial. It sets the stage for DNA polymerase by providing these essential primers. Imagine a construction site without a foundation. It's impossible to build, right? Similarly, without DNA primase, DNA polymerase wouldn't have a place to start building. But here comes a twist. Primers synthesized by DNA primase are made of RNA, not DNA. So how does DNA replication, which involves creating a DNA sequence, work with RNA primers? That's where another fascinating process comes into play, the replacement of these RNA primers with DNA. So, without DNA primase, DNA replication wouldn't even get off the ground. Now, you may ask, what happens to these RNA primers? You might be wondering, we're talking about DNA replication, so why are we using RNA primers? It's a great question, and the answer lies in the intricate dance of molecular biology. Primers, although short stretches of RNA, are the unsung heroes of DNA replication. They're like the warm-up act for a concert, getting the stage ready for the main event. They provide the necessary 3-hydroxyl group, acting as a starting point for DNA polymerase, the enzyme responsible for adding nucleotides to the growing DNA chain. However, there's a twist in the tale. As our story is about DNA replication, our RNA primers can't stay in the final product. So, what happens to these RNA primers? Well, they are replaced by DNA. During the elongation phase, another enzyme called DNA polymerase zips along the DNA strand, replacing these RNA primers with DNA nucleotides. This ensures that our final product is a perfect replica of the original DNA, with not a trace of RNA in sight. But remember, without these RNA primers setting the stage, DNA polymerase wouldn't be able to do its job. They are the unsung heroes, crucial to the process, even if they don't make it to the final curtain call. And that's how DNA primase, a seemingly small player, plays a massive role in the grand scheme of DNA replication.